and some reference. Next one. Okay, it looks like it's busy working. Yeah, it's busy working. Okay, so while it's busy downloading, okay, so when I was originally thinking of this course, I was thinking it would be nice to work on 2 different organisms, okay, but then time. Okay. The reason I want to work on 2 different organisms is because bacterial bioinformatics, when we are looking at the understanding, there's basically 2 things that we are trying to achieve with bacterial bioinformatics, okay, in the context of genomic epidemiology. There's lots that we can do when we're trying to understand bacteria in general, but in terms of what we're doing in a public health context, there's generally 2 things that we want to do, okay? The one thing that we want to do is understand the relatedness of isolates, okay? So the relatedness of samples to see if they are maybe evidence of transmission or not, okay? And the second thing that we're trying to do is to characterize isolates, okay? So what I mean by characterize is say things like what genomic features are present, especially we're interested in antibiotic resistance, but also which genomic markers of, for instance, lineage are present, okay? And then we're faced with a diversity of diversity of organisms, right? So who here has worked on Mycobacterium tuberculosis? Yeah, okay. Who here has worked on Klebsiella pneumoniae? Okay. So if we take Mycobacterium tuberculosis, and who here has worked on E. coli? E. coli in a disease context, enteric disease from E. coli. So these are 3 different organisms that have quite different biology. If we're taking Mycobacterium tuberculosis, okay, then it's a entirely clonal organism. It's not exchanging, there's no horizontal gene transfer going on in Mycobacterium tuberculosis. Each bacillus of Mycobacterium tuberculosis is a copy of the previous, except for whatever mutations, and these are typically point mutations. We're generally looking at point mutations. One particular one particular nucleotide changing to another nucleotide. Okay. If we're looking at Klebsiella pneumoniae, okay, the most of what we care about in terms of characterizing the organism, in terms of its antibiotic resistance, is arriving via horizontal gene transfer in the form of plasmids. So when we're looking at antibiotic-resistant Klebsiella, then it's normally because a plasmid has been transferred into the organism, okay? So if we look at the organisms, where did the pen go here? Yeah. So immediately we've got the question of core versus accessory genome, okay? So most bacteria are having the circular genome, but then we have plasmids in the case, and things like that, okay? Which is the so-called accessory genome. Then we've got clonal versus, I shall just call it non-clonal. It's a very wide category. Clonal versus non-clonal transfer of genetic material, okay? So when we say non-clonal, then we can include things like the operation of certain bacteriophages. So if we're looking at like Vibrio cholerae, the toxins that make Vibrio cholerae into toxigenic cholerae came in a bacteriophage, okay? If we're looking at many other bacterial species. We're talking about conjugation. We're talking about various forms of mobile genetic elements which can be within the core genome. We can also be talking about genetic material moving in the accessory genome of bacteria. Okay, and the biology of the organism is going to determine the analysis type that you use. Okay, because as I said, Mycobacterium tuberculosis, clonal, which means that we basically can talk about analyzing in terms of its ancestral reference genome and how it's changed since then. Okay. If we're looking at something like E. coli, the so-called core genome, in other words, the genome that is shared by all members of E. coli, Escherichia coli, it's only an estimated about 20% of the genes. A huge diversity in this organism. Okay, so the pathogenic E. coli O1H157, if I remember the numbers correctly, the estimated common ancestor between that E. coli and the K12, which is your typical lab E. coli that they use for biotechnology experiments and things like that, the estimated common ancestor was 4.5 million years ago. Okay, deep splits in the family tree of these things, and a lot of horizontal movement of things. Okay, so when we talk about E. coli, we cannot talk about one representative example of E. coli. And with Klebsiella pneumoniae and Bordetella pertussis, it's even worse because the main elements that we're trying to trace in an outbreak aren't even the core genome. It's accessory genes that are moving around plasmids and things like that, okay? So that gives us broadly two major approaches to understanding bacterial pathogens, okay? And the two major approaches, because in all cases we need to assemble a genome. We need a genome to study its diversity and things like that. And so we need assembly for everything. But is it reference-based, or is it de novo? Okay? And when we say reference-based, then what we mean is that we're going to choose a reference and then we're going to map reads to it with read mapping, and then we're going to call variants here, okay? So maybe over here, these reads are having difference to the reference genome, okay? And when we're doing de novo, then we are just taking the reads, finding the overlaps, and from there, building one or more genomic segments. Because of the limitations of the technology, often we cannot get a single finished genome from the reads, okay? Because of repetitive elements in the DNA and so on and so forth. And sometimes we are doing different things in different circumstances, right? Because if we want to look at the complete Vibrio cholerae, for instance, then the diversity is huge, okay? You get like so many different serotypes of Vibrio cholerae and all of those kind of things. But in the context of an outbreak, many bacterial outbreaks are clonal. So even if you've got a highly diverse species like E. coli, one particular outbreak, if you've got like so-called SCEC, cytotoxin encoding E. coli, then normally what you see is a sudden emergence of a cluster of cases that you want to trace back to a source. And then within that outbreak, then things are basically clonal expansion, even though the species as a whole has a lot of horizontal gene transfer. Okay, but unfortunately, in some species like Klebsiella, where what's going on is largely happening in the accessory genome, you can't even look at the clonal expansion of the core genome and look at your outbreaks that way. You have to look at the plasmids and so on. Okay, so how do we choose a reference genome? Okay, so in some cases, like again, Mycobacterium tuberculosis, Mycobacterium leprae or lepromatosa, then we have one reference genome that gets used for almost all analysis. H37RV in the case of Mycobacterium tuberculosis. Okay, and everything involves basically mapping reads to either H37RV or inferred ancestral genome that shares the coordinate scheme as H37RV. Okay, in the case of many other organisms, we have to go and choose a reference genome. Okay, the reference genome that I will be using today is one from early on in the outbreak of cholera in Haiti. Okay, so it's a sample from somewhere around 2010 was collected, was sequenced, a genome was assembled, a finished genome. Okay, so in other words, a single contig per chromosome. So a contig is one contiguous piece of DNA, and then that is used for analyzing like Haitian cholera strains. Okay. This is different from a lot of our African cholera analysis. We use a sample collected in Bangladesh in 1975. N16, I forget all the numbers, okay? But so if we look at the first cholera genome that was sequenced, it's the 1975 Bangladesh sample. There has been some later work after the original sequencing of Vibrio cholerae. There's been some later work to correct some likely misassemblies in the genome. But there's but the reason that people can do that with cholera is that since the emergence of the seventh cholera pandemic in about 1961, it has largely been a case of clonal expansion. Okay, it's now been one very successful organism that's like kind of grown from there. You can also there are tools that let you look for, if you want to look at an outbreak, look for a reference genome that is close to your read. So there's a tool called Reference Seeker that lets you go find in the databases one that's close to reads. But in general, especially if you're quite new to the field, I'd be very careful to choose your own reference genome. Rather work from what's been used in the literature, because you have to choose a good quality genome that doesn't have any misassemblies and so on, okay? So rather read the literature, see what people are using as a reference genome. But now in de novo, okay? So if you de novo assemble a genome, you've got a different set of problems, okay? So now, if you're going to do de novo assembly, you're going to get one assembled genome per isolate that you've sequenced. So how do you then compare them? Okay, you can do genome to genome comparison, but generally what is then used, instead of doing genome to genome comparison, what we then do is we look at the complement of alleles that we see in the genome. Okay, so using a system called MLST or CG-MLST, okay? So what that means is that across the genome of the organism, we've identified a certain number of loci where there are genes, and these loci, if we say CG MLST, it means, so the term MLST started with the traditional MLST of about 7 genes. You find 7 marker genes that allow you to distinguish one species from another, okay? And that for these particular genetic loci, then this particular pattern of alleles lets you know that this is this species and this sequence type of the species, okay? But with CG MLST, you look at the core genes, the genes which are present across the whole species. And then you look at the pattern, and each particular pattern gets associated a number and then if your sample is now having this allele, that allele, that allele, and that allele, then you will get a profile with the numbers of the alleles. So that sample, your isolate is now, for instance, 2, 1, 3, 3, okay? And 2133 identifies this sequence type of your isolate. So if you find another 2133, then you know that it's got the same genetic patterns at those loci in the sample. So you can look for these loci, even though your assembly might not have produced a single contiguous piece of DNA for the whole of your genome, you can go find these particular alleles within the different parts of your assembly, and you can get a profile, okay, using a service like PubMLST or Interrobase or similar, okay, depending on which species you're looking at. These different databases have these different what we call CG-MLST schemes. And then if you find another sample that has the pattern 2131, then we can say that there's a certain distance between this sample and that sample. And what is the distance between 2133 and 2131? We've got all these numbers, and what does it mean in terms of that distance? Number of different alleles, not number of mutations, because we don't actually know how many. So we don't know between allele 1 and allele 2, which is in the databases. We don't know how many mutations there are. There could be 5, there could be 2, there could be 1. Okay. So the difference here is the number of alleles difference, which here, that difference is 1, okay? This is 1 allele, because these are all the same except for 1, okay? So then what we can do is we can cluster things. So in this case, with the reference genome, we typically have either small insertions and deletions, but we focus on the single nucleotide differences, the SNPs, okay? In the allele-based scheme, we focus on how many alleles are different, and then we can just cluster. And we cannot talk about a genetic distance because we don't know how many mutations are different between the different samples, but we do know how many alleles are different, and then we can cluster. It's just that in both cases, we can talk about similarity and difference, but the measure is different, okay? Now, yeah, go ahead. Yeah. No, marker gene databases is more similar to this. Because your marker gene database is going to say that these GYRB, XYZ, so which genomic pattern do you have at this, so if we look at, for instance, so classic MLST, I think they've got it illustrated. So this is the pub MLST page for Vibrio, the Vibrio species, Vibrio genus, okay? And if we look at typing, just if I can. And if we're just using MLST, not CG MLST, okay, then we get asked for GYRB, PI, PYRH, REC-A, and ATP-A, okay? So these are the marker genes for Vibrio species. And then we can look what alleles we have, and then we can translate into allele numbers at those positions, and from there we get a sequence type, an ST, okay? And that will allow us to identify, but you see there's only 4 genes here, so the identification is fairly broad, you know? It doesn't, the CG-MLST for Vibrio, for Vibrio cholerae has, I think, about 2,000 alleles, so then you can get much closer. So for instance, in the Listeria outbreak in South Africa 2017-2018 was analyzed using CG-MLST of Listeria isolates, okay? And that used a scheme with 1,700 different loci in the scheme, okay? So you looked at the allelic pattern at those 1,700 positions, and then you got a profile. And then the final source was identified when a sample collected in the environment in a meat processing plant was the same as the profile collected in a patient up to, I think it was all except for, I think there was 3 alleles that were different between the patient isolates and the meat processing plant. And that's very close, you know, 3 positions. So, but one problem with this is that this system of numbers is specific to the different sites, right? So pub MLST might call a particular, you know, so we don't know what are the, if we look at, it's not easy for me to show you now, but if we look at like the pattern, so we might, pub MLST might say that ATACG is number 1, okay? Interrobite base might say that number 1 is ATTCC. You know, there's no standardization over how to number the alleles in CG MLST schemes, okay? Which means that if you use the Institut Pasteur, BigsDB server, if you use Interrobase, if you use PubMLST, if you use cgmlst.org, you all are going to get different and not compatible results. You have to always get your sequence type from the same typing scheme. You know, they're not comparable between each other, okay? Which, yeah, it's a weakness of the system, okay? There are some discussions as to how to overcome it, replace it with some other interoperable system, but that's where we are at the moment. Okay, let me see if my download is finished. Yes?
The 2 assembly, the reference I gave it, right? That's the novel. How does it really work? I'm trying to
So how does it? So how does it really work?
Yeah.
Okay. So I think when you're dealing with SARS-CoV-2, you've been exposed to reference-based assembly. Okay. What de novo does is that it finds the overlaps between the reads. Okay. Typically using a tool like Shovel or Sketcher or Spades. You know. So these are some of the tools that you use. So it reads. So you first, everything starts with quality trimming, right? So you remove the low quality reads. The low quality parts of the reads, okay? then what you do is you find the overlaps between the reads, typically with short reads using an approach called the brain graph. And then you stitch together the reads. So that if this one's overlapping with that one, and then it's overlapping with the next one, you find a path to all the reads that allows you to stitch together a bigger chunk, okay? And so let me just do a quick intro to like how that works. So it's the equivalent of if you took a book, you photocopied the book lots of times. That's your PCR, making copies of the book. Now you run it through a shredder. Now you're trying to reassemble it, okay? The only problem is that if your genome has repetitive elements in it, okay? So let's imagine that this piece has the same sequence as this piece, okay? Then, if you've got a read over here, right? And it's got part of this piece. Remember, the read is going to look like, it's going to have that little red bit like from there, okay? And then, on this side, we have a read over here. And it has that red bit. Remember, the red bits have identical sequence data, okay? Because these are repetitive parts of the genome. So it's going to look like you can put the two together based on that repetitive piece, and you're going to get a misassembly because it'll pull the two repeats together, okay? And so you can have misassemblies, but the assemblers tend to like break up these misassemblies, but the end result is that you can't decide where a read should end up. So instead of getting one a single thing back, let me find the diagram that I want to use for this. I wanted to make some slides on this, but then my computer crashed. So yeah. There's a tool called Bandage that you can use to assess the shape of assemblies. Let me just find the picture. This is the wrong page. This page, where did it go? Yeah. Then. This is a assembly graph of Staphylococcus aureus genome done in Galaxy using only short reads. And what this assembly graph shows is that like this is the part of the genome, and then over here it can be either this piece or that piece, right? So when we've got a disagreement in the reads, then maybe it's this way, maybe it's that way, okay? But where these things touch, it's typically where there's a repetitive DNA. We've got like a chunk that's all unique, and then if we look at what this means in terms of the assembly graph, okay? Then what we can see here is a piece of genome, and then it's either followed by this piece or it's followed by that piece. So that's why we end up with these weird splits in the assembly graph, because we're not able to resolve the repeats with short reads, because short reads are not long, they're not covering the whole repeat, okay? What we would like to see if my mouse can operate. is that pretty much one circular chromosome, okay? And this is done using nanopore reads, okay? But before you get very excited about nanopore reads, the nanopore reads allow us to generally cross across the repetitive regions and assemble things together. But until recently, if you look closely at this assembly, you would find that there's mistakes at the small level because nanopore is not very accurate. So the individual bases inside of here, the structure might be right, but the individual bases might be wrong. Okay? There's a guy called Ryan Wick, no relative of John Wick as far as I know, who is in Australia, and he does a lot of evaluation of assembling from nanopore data. And so he's basically updating his results every few months or whenever the new data is coming out. Like now we've got the R10 flow cells, Kit 14 chemistry, and all of those kind of things. And he's redoing this stuff. So, and then he's publishing his results and his conclusion that so what you can do is if you've got nanopore and Illumina, you can use the Illumina to correct the individual errors within your nanopore assembly. That's a process called polishing, okay? But of course that means you must have both the same isolate sequence with Illumina and with nanopore, okay? But there was Ryan and co just came up with a new assessment. They also said that you must have at least 25 read coverage on a position to trust polishing. If you've got less than 25 reads coverage, you can actually introduce errors in the polishing. Okay. The other thing is that how much do you need polishing? The latest results from Ryan are showing that genomes can get to within, less than 20 mistakes in the whole genome at this point with the latest nanopore data. Okay, I think he was using a Klebsiella genome for that. But the other thing is that your base calling, the base callers are that you get from they've been getting better and better. We are on what we are on now. Dorado, I think, is the latest base caller. Is it Dorado? They all named after fish. What was it? It was Guppy, and then they came Bonito, and now there's Dorado. Okay, so now we're on Dorado. Okay, I think it's the latest one, and they're giving better accuracy as they develop, but within the base callers, if you they have a model, and that model is tuned on typically tuned on human DNA. So if you get a model that's specifically tuned for bacterial DNA, it can actually increase the accuracy even further. And there has been some work on tuning base caller models for bacterial DNA. And then the final thing about nanopore accuracy that's showing up in the results is that what's probably throwing off that last bit of accuracy that we want is methylation of the genome. Because remember, as opposed to Illumina is working with a copy of your DNA, okay? So when you're making Illumina library, it's not the original DNA that you extracted. But when you're working with nanopore, you are working with the actual original extracted DNA, and the DNA has the methylation of the original DNA, and that methylation can throw the base caller off, okay? So some of the areas where it's difficult to get accurate results seem to be related to methylation patterns. So, yeah. Anyway, so ideally we have all of this, but what's this thing over here on the side? Okay. Great, nice circular chromosome, nearly end-to-end complete. Okay. Is that a plasmid? Maybe. Is that something that you couldn't fit in? So the other difficulty with assembling is that when you are resolving those bubbles, okay, we're seeing those like like the DNA could go this way or that way, okay? Some assembly methods have not been tuned at assembling plasmids. Okay, so what gives you the best chromosomal assembly, those settings might not give you the best plasmid assembly. That's still an ongoing area of investigation, because worst case scenario, you eliminate all bubbles, okay? So you try and make it all fit into one thing, and you actually eliminate plasmids entirely. There have been some examples of optimizations going in that direction, okay? But as you can hear from my description of these things, this is not press the button and go science at this point, okay? This is a whole field of very active research and investigation, okay? Which has to be explained to your colleagues in the lab that this is still we're going to have to. This is not a production project where you can just say we're going to do this. This is an area where we have to research, try methods, keep up to date with the latest developments and so on and so forth. Okay, it's very important for to have these discussions early on in a sequencing project, that you don't get people giving you or coming with expectations, and then they're like, okay, here's your data. Can you analyze for me, please? So anyway, so that's assembly, but I'm not going to start with assembly. I'm going to start with the easier problem of reference-based methods. Okay. Now, let's see. How are we doing for time? Let us start with cleaning our data. Okay. Yeah, I think so. Okay. Now we're going to have to deal with another bit of. My download is still progressing. Okay, so let me. There's 5 samples here. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm busy with the last. You should get 10 FASTQ files downloaded. Okay, you should have 10 FASTQ files downloaded. Has anybody got all of them yet? Mine isn't complete yet. Okay, so we're nearly ready to go. Okay, so the next thing is, I said we're going to deal with reference-guided processes. Okay, so what are we dealing with here? Okay, where do we want to end up? So I've spent a long time talking about CG, MLST, and loci and alleles and all of those kind of things. Okay? Let us leave behind the world of assembly and CG, MLST and consider reference. Okay? In a reference, each isolate, the genome of each isolate, you guys, I think, have all worked on SARS-CoV-2 analysis, so you've seen how this works. Okay? You will take the reference, you will identify for each sample the differences. So let's call this sample 1. Let's call this sample 2. Maybe some slightly different differences, okay? And then, where there is not a difference, where there's not a mutation, okay, you will copy the DNA from the reference, right? Because if there's no mutation, then it says that the DNA is the same as the reference for your chromosome, okay? Except, when will you put ends in the sequence? I'm going to throw this one away. It's done. When will you put ends in the sequence instead of copying from? When will you see ends in your final consensus sequence? And not if your name is base space, but that's another story. You guys have all done SARS-CoV-2 genomes. Do you get the whole thing with no ends? No. Then why are there ends in your SARS-CoV-2 sequence? Coverage, yes? Well, so got it there. If you don't have enough reads, and then you don't know whether what it is what was in the reference or not, because you can't call variance. If you can't call variance, you can't put anything there. You put an end there. So it's always variant calling and looking at depth. Variance and depth, okay? So you can recognize if you've got poor sequencing coverage by these ends. Don't do what base space, the Illumina tool, I don't know if they've fixed it yet, but for a while I could recognize SARS-CoV-2 genomes assembled by Illumina base space because they would always put reference even if there was no data, okay? And how could I recognize that? Because if I've got, let's say, an Omicron, okay? Then Omicron always has, let's say, this pattern of mutations. All Omicron has these mutations. But instead of putting, I don't have enough coverage there, so they would put the genome as if it's referenced at this position. Okay? Instead of putting, they should put ends, but they put reference. I did that once for a SARS-CoV-2 genome, and I'm still doing my atonement, and I'm still apologizing for having done that. One should never do that, okay? Because then Nextclade is going to tell you you've got a reversion. This mutation has gone back to ancestral, okay? And I literally got to the point where when people would come up and say to me, why does my SARS-CoV-2 genome get this warning message from Nextclade about reversions? I'd be like saying, you've been using Illumina base space, right? Because they didn't fix that bug for so long, and that was what was happening, okay? Which is also why I absolutely love it when I can actually see what your bioinformatics method is doing, okay? Because if I can't, then strange things happen and we have to go, wait for Illumina to fix it, but yeah. So this is the basic approach. The end result is that your final genomes all end up being the same length as the reference. Because we've taken the reference and we've just inserted, unless we're putting insertions and deletions in, but let's assume that we're putting just the single nucleotide changes in, okay? Great. All is perfect. How many chromosomes does Vibrio cholerae have? 2. The cholera people know. Vibrio cholerae has 2 chromosomes. Okay? Now what must I do? Because when I do my phylogenetics, I want to give it a stack of genomes that are all aligned to each other, like this. But what do I do if I've got 2 chromosomes? Well, one thing I can do is do the one and do the other and have 2 different phylogenetic trees. Don't really want to do that. What I do is I mash them together. So this is chromosome 1, this is chromosome 2. And then I map the reads. I map the reads against my combined chromosome. Okay? Well, how can I do this? Why can I do this? Why is this not a heresy? Because I'm still collecting the genetic diversity of the 2 things together, okay? So I use the tool called, I'll give the notes later when I get a chance to writing it up. I use the tool from the MBUS toolkit to match the 2 things together. But my colleague Kai, there's other tools for it. I'll make notes as to how I got this thing. But so I took these 2 files, NC016446 and 445, and I mashed them together to get this, okay? And if we add together the sizes 7 and 7.5 and 2.5, they get to approximately 10. So this one is those two combined. Okay? Now, .genbank. Okay? What does .genbank mean? It means I got it from the NCBI GenBank database. Okay? And this is showing me, and this is useful to me, and I will show you what we can see inside these files. These are not FASTA files. Sorry, I must go into the Vibrio folder and download it to here. More. These are not FASTA files. GenBank files contain both the sequence and information about features on the sequence. So, for instance, if we look here, we can see that at positions 539 to 36 on the reverse strand, there's a gene called 4K with a locus tag of what, okay? The reason I'm using GenBank files is because later on I'm going to use Snippy for calling mutations, okay? And if you feed GenBank files to Snippy, it not only tells you where the mutations are, but it uses the annotation to tell you what the effect of the mutation is, whether it's an amino acid change or not. But we'll get to that later. Okay. Now, it's time to get to play with dangerous things, okay? Which is the actual code of these things. First thing that we need to do, we need to prepare our Bactopia to be ready to be used, okay? I don't think it's going to affect us so much, but let's just run, okay. We've now all got a Bactopia Conda environment. So how do I activate a Conda environment? Conda activates the name of the environment, okay? And if I can just run Bactopia, then it tells me it can do various things. Okay? That's the Bactopia command. Normally, the first thing that you ever do after you've installed Bactopia is that you run Bactopia datasets. And this goes, oops. Don't worry about the messages, that's because I've run this command before. But if you say Bactopia datasets, then it downloads the databases that Bactopia uses. Okay, so this is a few different databases. It'll take a while to run when you run it the first time, okay? But I want to take a digression, okay? I said I like knowing what Bactopia does, okay? So Bactopia is a shell script that wraps around a Nextflow pipeline. What I said to John the other day about Nextflow is that yes, understanding how to write Nextflow can be challenging, but what I wanted you guys to pick up from Nextflow is how to read Nextflow, okay? And specifically a Nextflow project that follows the NF core standards. So the first step that we're going to do, and I'm going to try and get there before lunch, is that we're going to do quality control with Bactopia, okay? And we're going to use a sub-workflow of Bactopia called Clean Your Reads, okay? So let's go and try and figure out how Bactopia works. And we're going to do that by reading the code. Bactopia GitHub. So GitHub is the source code website where you find the Bactopia code. Okay. Bactopia dataset should take quite a while to run for people because it's downloading various things. Is Bactopia dataset working? Is it downloading things for you? No, is it not? What is it doing when you try Bactopia datasets? Is your Bactopia working at all? Okay, yours is done. Okay. So let's go read some code, okay? So this is the Bactopia source code on GitHub. So if we look at the Bactopia source code, if I just get everybody's attention for a bit, okay? So the first thing that, by the way, that we notice from the Bactopia source code is that the most recent person to make changes to the source code is somebody called Arbati, okay? I don't know, Americans, I don't know if he pronounces his surname like that. Robert Petit, that's the guy who wrote Bactopia. But it follows the structure of a NF Core Nextflow project. What I mean by that is notice these folders called workflows, sub-workflows, and modules. Okay? So, Bactopia has multiple workflows for doing different tasks. If you read the Bactopia main documentation page, Bactopia, then it tells you about various things that it can do, and there's Bactopia tools and all of those kind of things. Okay? And I want to look at a workflow called clean your reads. Okay? But I want to do this as an exercise in reading Nextflow. This screen is still not big enough. If we look at what the code looks like at the top, then we see it says Nextflow enable DSL2. So this is a DSL2 Nextflow workflow. Okay. Then it talks about some config files. We can skip over the config file section. It talks about validating the inputs. We can skip over that for now. I want to focus on what happens next. It says import local workflows, okay? So it says here include gather from, and there's a name, include QC from, and there's a name, include scrubber from, and there's a name. So it's got three different modules that it's importing called gather, QC, and scrubber, okay? And the names give you a bit of an idea of what they do. Okay, so if you read the Bactopia documentation, then one of the things that you'll see is that you can actually tell Bactopia to download data from the internet. Okay, so what Gather does is it prepares your data ready for analysis. Okay, QC, quality control, and then Scrubber. So let's see what it does with these things. Okay. And here's the main workflow, clean your reads. And it says here, line 58, remove host reads. Okay? So, if you read like the code, this is the main workflow, and we just focus on the things that use those modules that it imported. We see line 54, okay? Gather, and then it basically does a whole lot of things, and it gets a channel back, okay? And then, line 57, if parameters says enable scrubber, then the comment on line 58 says remove the host reads, or the human reads, okay? And then run QC. And if you don't enable scrubber, then run QC anyway. And then it basically gets the versions, and then eventually it runs a thing called dump software versions, okay? So my point about this is that reading this thing, we can just, even if we don't understand exactly what it's doing, we can see, and then down here at the bottom, the workflow ends. Even if we don't understand exactly what it's doing, we can see that basically the clean your reads workflow is optionally, depending on whether you've enabled it, removing host reads, and then it's running some quality control. Okay? So now, we said that we saw that QC was imported from sub-workflows local QC. So if we go back into the main code, and we go find sub-workflows local, and there's so many of them, but we were looking for something called QC. Where's QC gone? There's QC. And then main. And then there's the workflow QC. Okay? And then it uses a QC module, which is running from modules. You can follow it further. It says that the QC module comes from the modules directly. Modules local back to up here QC. Okay? So this thing, QC, the sub workflow, is just running a module called QC. Okay? So let's keep chasing the definition of things. So it says that that is in modules local QC. Let's go find modules. And then local. So the NF core modules are the ones imported from the NF core project, okay? But local are ones that Robert wrote himself. Oh, there's QC. And there's main. Okay? And there's process QC. If we look at the bottom, there should be a workflow. Is there a workflow? There's a lot going on here. Yeah, so it's just got a process, which is the process in Nextflow. Remember, the thing that does the actual work. Okay? So this one is more complicated to read. Process, a whole lot of inputs and outputs, and here's a script, okay? Lots of stuff, more lots of stuff. And then we start seeing code. Line 60. This is shell code. If the run type is hybrid or the run type is short polish, then enable line 61, ONT, and enable Illumina. So enable both. Okay? So that's because hybrid means a mixture of nanopore and Illumina, and short polish means polish with short reads. So if that's the kind of workflow, if that's the kind of run that we're going to do, then enable both of those things and tell it that, and line 63, say it is hybrid. But if the run type is ONT, then enable ONT. If the run type, otherwise enable Illumina, okay? Then is assembly, you can include pre-assembled data. So if it's assembly, then just copy that across. That's not what we're dealing with, okay? If we're going to skip QC, if the skip QC is true, then it just copies things to the final destination. You see, it's just copying this and copying that. It's not actually running anything, okay? Else, so now we're not skipping QC. If ONT is enabled, here we get into what the quality control procedure for nanopore reads with this tool is. What tools is this? What tools is Bactopia using for quality control of nanopore reads? Pore chop, yeah, which is removing the adapters if necessary. And there's a thing called NanoQ. You know, so with the quality filter, and it's NanoQ is taking, so I'm used to for nanopore reads using a tool called Filter Long, but it's kind of doing the same thing. It's saying minimum length and minimum quality. So Filter Long, it does the same thing. It says basically, I want to throw away very short reads and I want to throw away very low quality reads. But NanoQ is doing the same thing. I've never heard of NanoQ, but just reading the code, I can get some idea as to what's happening here, right? Okay, so that's if you're doing quality control on nanopore. Else, if we're doing Illumina, what is it doing? First, line 105, if we're using BBMap, then it uses a thing called BBMap to clean the reads, okay? And it's doing some stuff to make sure that paired end reads have matching IDs, and then repair more stuff. It's unzipping some stuff over here. It's converting compressed to uncompressed, and then finally it's running there. BB duct to do the cleaning. Okay, so this is all if we've enabled BB duct. That's not the default. Okay, and then doing more BB duct to remove FireX and all of those kind of things. Okay. Else, if we don't enable BB duct, yeah, Stanislav was just telling us, what is this thing using for quality control of Illumina reads? FastP, that we've heard about the other day, right? So, and then we get down to the end. And then there's some stuff about error correction. And finally, if you want to, there's an option about if the error is nothing. So here, if the error is equal to 0, in other words, it's only continuing if there's no errors thus far, okay? Then it's using different commands. So sometimes you don't want to have like massively high coverage on your genome. So optionally is reducing the coverage to a maximum amount, okay? And then lots more stuff. And finally, it compresses your reads. If it's uncompressed reads, it makes it into compressed reads. Pi GZ is parallel gzip. Okay, so it's just gzip that you've seen before for doing compression, but a parallel version. Okay, and then it does some various things with it. And finally, it does some quality stats before and after QC, using fast QC, nanoplots if we are doing nanopore. And then there's a final QC check, and I can't see what it's doing here. I think it's doing some other stuff. And I'm not going to read through all of that. But my point is that this is a long, complicated script. But without having to read every bit of it, you can see that like quality control for nanopore is this thing called NanoQ. For Illumina, it's either BB duct if you've enabled that, or FastP. And then it will do this thing, and then it will end. Okay? So, where did it get the software from? Okay? At the top here, it says two things. In the process, I don't think you've seen this yet in the next flow, but I can't remember. Lines 15 and 16, okay? It's either using Conda, okay? Or it's using containers, okay? And if it's using Conda, it will create the Conda environment which is defined up here somewhere in line 9. It will create a Conda environment with the tools that you need. And if you're using containers, it will let you either use Singularity or Docker containers, okay? Now I've tested these tools with all 3 options. My experience is that the Conda is error prone. The Conda, for this tool, it actually works. But for other of the Backtopia workflows, it has problems finding all the necessary things with Conda, okay? Then I tested it with Docker and with Singularity, and both of those container systems work. They download the containers and they run the software from the containers, okay? So, to me, if you're running things on an individual machine, I find that Docker, if you can use it, is the easiest system to use, okay? Simply because there's something actually quite interesting here. Line 17. If we're using Singularity, okay, then download a Singularity container from, do you see what it says here? The Nextflow people use resources built by the Galaxy community to get their containers. And my experience with this website where they download Singularity containers is that it's kind of slow, okay? Because it's a free project, you know, with just one server and all of those things. I have pre-downloaded all of the Singularity containers, if anybody wants the relevant ones, okay? But then this key.io, this is one of the big Docker container repositories, and it has a lot, you know, it's used to lots of people downloading things. So it's, in my experience, the Docker downloads are faster than the Singularity downloads, okay? But it depends on what in your environment, whether you've got Docker, whether you've got Singularity, okay? So yeah.
Does that happen after installation?
That happens when you run the workflow for the first time. So and that's actually, I mean, Panga's question is a very interesting one, right? So when we run the workflow for the first time, it downloads things, but not all of us have great internet connections, okay? So one of the things that, so in last night's Phage infrastructure working group meeting, I'm working on a blog post for Phage on how to pre-download these things so that, because, you know, so that it's ready before you even run the workflow, okay? So I found some scripts and things like that, and I find that this is quite useful because then you know that it's going to run because you've got the software already available, okay? It gets more complicated when you are using clusters, okay? If you've got one computer, then you're downloading the software to one computer and it's downloaded and it's fine. But what happens if you have a cluster with 3 different worker nodes, for instance, or something like that? Then if it's downloaded to the one computer, it might not have the software in the other computer. Also, what happens if Stanislav and Islamia fire the same workflow off at the same time, and it starts downloading 2 things at the same time, you know, and using double the bandwidth and things like that. So, so the best way to make your environment ready for running this workflow, it's still something that we're busy discussing in the infrastructure working group, but some way of doing it. So the one option that I found is to identify which containers you want to download and download those ones ahead of time, okay? The other thing that I've tried is take a small sample and run a small sample through the workflow so that it does all of the downloading of containers and things like that, and it's then ready for when you want to use it, okay? I think the easiest that we can do for the people who are using Linux or who are using Windows with WSL, I think probably the easiest option is going to be to use Singularity containers, because it's easy for me to download them to people. But I'm still, yeah, we will see, I think, after lunch when we get going with this. I think it's going to take us most of the afternoon to get everybody's computers set up to do this, okay? The one good piece of news with this thing is for those people with the Macs that are M1, there is a way to use Backtopia without using Conda, okay? Because you can use it from Nextflow directly, but I will get to that once I've got all the other stuff set up. And then Docker can work even if the other things on the Mac are not working. And George, actually, it might be an interesting experiment for you as well because your M1 Mac comes with Docker, right? You can run Docker on there, right? And Docker can use, is it called Rosetta, to translate things on the fly? So you should actually be able to run Backtopia, Nextflow workflows like Backtopia on your M1 Mac, doing stuff that you can't do because Conda doesn't work. And how much RAM you got?
16 GB.
Yeah, so you should be fine. And your M1, how much RAM? 8. We'll see. It should still work, but it's going to be an interesting experiment.
Yeah, we can try to do that one.
Yeah, and in fact, yeah, no, I want to try with the M1 people, but I want to get the other things sorted out first. Okay, so it's after lunch. We're going to actually get everybody like running the Backtopia and doing the first step of the QC. And what I want to get done today and tomorrow is from the QC also do the reference-based sorting out the genomes and the phylogeny from there. That works quite easily once you've got your basic environment set up, okay? But I see that there were some problems with the Backtopia dataset commands. So if anybody still, I think we can go for lunch, but if anybody, okay, firstly, if there's any questions about any of this stuff, because I've gone over a lot of stuff, please ask them. And if anybody still has computer problems, then please let me know so that I can try and fix them during lunch, okay? Before I create a whole new set of computer problems after lunch. What? Do you have a connection problem? You don't have internet? Discrimination.
There was something I think it might have been before that.
Conda activate Backtopia. I just ran Conda activate Backtopia and then Backtopia datasets. Okay, let's just go to the bathroom quickly. But
Yeah, just like
then I'll come help people with their errors.